Hi, Michelle Glass here, and welcome back to another episode of our Chapter 3, Lectures 3 series. And we're continuing our discussion here of our organic macromolecules or biomolecules. And our topic for this video are the lipids. The lipids include fats, oils, and waxes. And the key difference is when you're comparing these three types of lipids at room temperature, you'll see that fats are semi-solid. So think about butter and how it, holds its shape, but it's spreadable at room temperature. And then oils would be, of course, liquids. I think most of us have a pretty good impression of those. And waxes are solid at room temperature, but of course can melt. So think here about a candle is probably the easiest association to make there, or crayons. So you're all familiar with these three different types. Now lipids, as we look at the lipid structure, it does not have a true monomer. So previously, when we were looking at the carbohydrates, we studied that the monomer was the monosaccharide, and I mentioned that as we look at each of our macromolecules, we do want to be familiar with the building blocks, but we really don't have anything to list here for our lipids. We will see that our lipid structure is described as a long hydrocarbon chain. And here the hydro means hydrogen. So I've drawn an example here of a long hydrocarbon chain. The ratio here is usually 1 carbon for 2 hydrogens, and then there are oxygens involved here. So you have a carboxylic acid group here. In my drawings, in my laser pointer. So here, just like with carbohydrates, we have carbon and we have hydrogen and we have oxygen, but our ratio of carbon and hydrogen is greater than our amount of oxygen that we see in this molecule. Because of this long hydrocarbon chain, our lipids are going to be described as hydrophobic. Hydrophobic means water-fearing. So we're going to see that, and you know this already, oil and water don't mix. They don't mix because oil is a lipid, or lipids are non-polar because of that long hydrocarbon chain. And so we can use the term nonpolar, or we can talk about them as being hydrophobic. That's going to be really significant as we start looking at the cell structure. So we'll be talking about this quite a lot. The main function of our lipids we can consider to be energy source. So when we're talking about our cells making energy, we see that they use carbohydrates and they use lipids as their main sources of energy. Typically, we think of our carbohydrate as the quick energy source, so that's kind of the first go-to for your cells, and then the more long-term energy source are the lipids. So we do see this as another important source for our cells. There are some different types of lipids for us to talk about, not just the categories, fats, oils, and waxes, but at the molecular level. So we have a group of lipids called glycerides. Glycerides are characterized by having a single glycerol and at least one fatty acid attached. So we have a glycerol and a fatty acid attached to make a glyceride. Notice that there's room for additional fatty acids on this glycerol. So we have what we can call a triglyceride, which is something that you probably have heard before. This is listed on nutrient labels. So tri means 3. So in a triglyceride, you have 3 fatty acids, 1, 2, 3, paired to a glycerol. For animals, this is how we store our fat. So when we're talking about fat tissue, those cells are full of the triglyceride molecule. So this is how we store extra fatty acids from our diet. And that can be important for our bodies because not only is it providing an energy source, but it's also providing a layer of insulation. So that's important for our bodies. Triglycerides are also making some pad around some of our key organ structures, providing some shock absorption, so some protection that way. We'll see fat pads in our movable synovial joints so that you don't have bone to bone rubbing against each other. So we'll see these triglycerides having lots of different types of functions in our bodies. Now, as we're looking at this triglyceride, I have built it using what we can call saturated fatty acids. A saturated fatty acid has all single bonds because it is saturated with hydrogen, meaning these carbons, remember, can make 4 bonds. So we see each one of them is paired to another carbon and then 2 hydrogens as well. So this is an H2, just to kind of save room there instead of drawing all of my bonds. So saturated fatty acids have single bonds. They are saturated with hydrogen atom, and this makes them solids or semi-solid at room temperature. So animal fats are saturated fatty acids. So butter, the fat that we have in our body, would be saturated fatty acids. We also can see what are called unsaturated fatty acids. And to be unsaturated, you don't have as many hydrogen atoms, but you still have to complete the bonds of carbon, and you do that with double bonds. So here I've drawn 1 double bond. I could have 2, I could have 3, I could have more double bonds. The significance of those double bonds is it makes a little kink or bend in the molecule, and then this actually changes the property of the lipid. So instead of it being semi-solid, it's now going to be an oil. So we see plants and fish store their fat in the form of oils, and so they're unsaturated fatty acids. So for plants, you typically see these oils stored in seeds, so little compact versions of the plant. Not so much in the rest of the plant's body. Typically, plants are storing their energy in the source of a starch, right? Because it's easier for plants to store. And humans and other land animals, we don't want our bodies to be too slushy, storing all of that lipid as oil. So that would be the significance of having this semi-solid fat. Now, in the laboratory, you can hydrogenate an oil. So you can take an unsaturated fat. So you can take something from the plant or the fish that might be easier for you to obtain, cheaper to process, and maybe somewhat healthier. And you can make a spreadable. You can turn it into a semi-solid by adding hydrogen. So hydrogenation just means addition of hydrogen. So what you're doing there is you're taking that unsaturated fat and you're breaking that double bond and you're adding hydrogen there, so you're converting it into a saturated fat. Now, one of the byproducts of this process is making fatty acids that are called trans fat. And the trans here is talking about the orientation of the molecule. And these don't actually work the same way in our bodies. These are actually found to increase the risk of heart attack really significantly, to the point that the FDA is banning these from our foods. So these have been removed from our foods and no longer, hopefully causing a problem for us. Now, lipids are not only important as energy sources, but we also see them involved in the structure of our cell membranes. And cell membranes are a universal feature of cells. So again, whether we're in domain archaea, bacteria, or eukarya, we will see cell membranes there. And these cell membranes all have the same basic structure. One of the significant molecules of your cell membrane is the phospholipid molecule, and we'll have, an entire little talk just over that phospholipid. And then we'll see cholesterol is another important component of your cell membrane. When we look at our phospholipids, we'll see that it's the phospholipid itself that's creating what we will call a semi-permeable structure to the cell membrane. Semi-permeable means some stuff can penetrate and some stuff can't. Now, the significance of the phospholipid molecule is that it will have what we call a hydrophilic head and then a hydrophobic tail. So that means it has a water-loving head and a fatty acid water-fearing tail. And so this will be important. We'll talk about it again in the future. So just keep that in mind. The cholesterol molecule is important in stabilizing that cell membrane. So it's helping to keep it from being too fluid. We'll see also that cholesterol can be used by animals to produce steroid chemicals. Steroids, you probably know, are hormones. Hormones are little communicating molecules that move through the bloodstream. Some examples of steroids would include your sex hormones. So these would be produced either by the ovary, estrogens and progesterones, or produced by the testes, which would be testosterone. There are some applications of using steroids medicinally. We can actually create steroids in the lab. So we have what are called anabolic steroids. Anabolic steroids are a synthetic form of testosterone. They're called anabolic because anabolic reactions are building reactions. And if you see, some of the jobs of testosterone are to increase your muscle mass, increase your bone mass. And then testosterone is also responsible for what we can describe as the male secondary sex characteristics. Secondary sex characteristics are those things that start to show up at puberty that make the adult male body look different from the adult female body. And so testosterone is really helping to kind of modulate those activities. So if you administer anabolic steroids, you still get the increase of the muscle mass, you still get the increase of the bone mass, and you're still getting some of those secondary sex characteristics. So we can use anabolic steroids to treat some diseases that destroy muscle to help increase the production of that muscle. And some athletes will use anabolic steroids as a way to increase muscle mass, right? If you're abusing the anabolic steroid, there are lots of different risks associated with that. So things that you've heard of before, like roid rage, dramatic mood swings, it can trigger depression. It can also lead to liver damage or liver cancer. You can also see an increase in your cholesterol production. You can also see an increase in your blood pressure. And then because you're flooding the body with this testosterone, then basically the testes, if you're male, will stop producing testosterone. And so then you actually see kind of like a reversal of those secondary sex characteristics. So it can lead to shrunken testicles and a decrease of the sex drive and infertility and formation of breasts and lots of other undesirable characteristics. And that is it for now.